My name is Tom Freeman, and I have the privilege today of interviewing Professor Anders Bjorklund from the University of Lund in Sweden. He is an accomplished scientist who, by the age of 47 years old, has completed an MD PhD at the age of 24, and he now has over 400 publications, including 282 original articles. Of course, the reason that we're interviewing him today is because he's considered by many to be the father of the field of neural transplantation. He gave the keynote address here at the 61st annual meeting of the American Academy of Neurological Surgeons in Boston on the use of neural transplantation for therapeutic purposes, with a focus on Parkinson's disease, but also reviewing a number of the other possible areas of application of this very interesting technique. And I'd like to start off by welcoming you here, and also to get a little bit of historic perspective on the University of Lund where you trained.
It has a good neuroscience tradition, which I think is important in this context, particularly in the field of Parkinson's disease. Arvid Carlsson, who is one of the critical figures in the early discoveries of dopamine in the brain and development of L-dopa as a therapy. He started off his work in Lund in the 50s and made some of his critical discoveries there. This was, or gave the impetus for Hillarp and Falk, who was working in my histology department, to look for a method to study these compounds, the catecholamines, on the microscopic level. And this gave rise to the histofluorescence method that bears their name, the Falk-Hillarp method, which was introduced in 1962. And that method has had a wide impact because it gave a totally new angle on the studies of transmitter-related systems in general, because it was the first method that allowed studies of neurotransmitters at the neuronal level. And secondly, also for studies of the catecholamine systems in the brain. And when I started in the department in '66, the method was still quite new, and it was harvesting time. There was everything new that was looked at gave new discoveries, new data. Publication rate was high, and it was a very dynamic lab, and then great fun for a young person to join. So I was put at studying the hypothalamus because of Falk's endocrine interests. And the issue then was neuroanatomy, to study the organization of the catecholamine systems in the brain, which was incompletely known. And in this way I got to learn neuroanatomy, neuropharmacology, techniques, a number of microscopic techniques I got good exercise in. So it was a very good way to start.
My understanding is that your direct supervisor was Professor Falk. I wonder if you could tell me a little bit about how he trained you and some of the ways he taught you and honed your thought process and how he influenced you.
Yeah, he maintained he understood nothing of the brain, which gave me gradually more and more freedom, because he felt that he would be doing better by not intervening too much. He had his great talent was in a kind of administrative sense, or is, I should say. He's just about to retire now from his chairmanship, and he he was gifted in generating money to get people together, to get them to work together, and a very inspiring type of personality. And I think it was we missed very much when he decided in the 70s to go into more administrative duties, and then finally he's ended up in a different field in dermatology later. But during the first years of my work in the department, I had very much positive input from him. And when he left his lab interest, I was probably sufficiently dry behind the ears to want to do my things myself anyway.
And then I guess Professor Hillarp became the chairman in Stockholm and trained a group of neuroscientists there. And there ended up being two very distinguished groups in parallel. And
that's right.
What if you comment on
he
the
moved in
relationship?
he moved in 62, very soon after the method was discovered, because he was recruited to the chairmanship in histology at Karolinska Institutet. And there he gathered a very active and very gifted group of people. And for a few years there was fierce competition between the two groups. I didn't experience that very much, because when I started, I think most of the conflicts that generated had started to disappear. But for a few years it was not just competition, but conflict generating, which was unfortunate. On my generation, though, the those who were trained in that period, we've found that we can live very well together. There's a healthy competition. We have always competed, but in a friendly way. And I think that that's been very stimulating for both the Stockholm group and for us. So I wouldn't like to miss that.
When I was reading over your curriculum vitae, something popped out to me that I thought was very interesting, and it's a pattern very similar to what I've seen in my limited experience of talking to other very distinguished scientists, including many who have won Nobel Prizes or the Lasker Award or have been equally distinguished. And that is that the personal stories I've heard of people that I've respected tremendously has been quite similar. Even though the exact stories are different, the pattern was very similar in that many of them started working in laboratories of very distinguished scientists, got a very interesting start, and then followed a very natural pathway from one idea to the next and pursued the interesting observations in depth that ultimately brought them to an area that had nothing to do with where they started. And when you go back and ask, they say, well, yeah, I really never had that idea of where to go, and I just sort of followed my ideas where they took me. And your career started off really in an area of mapping, and then you've had a second career that really started in 1971 with regeneration and plasticity, and then in 1976 it moved into neural transplantation. That's three really distinguished and separate careers. And I would just hope that you could sort of take us through how you got from one area to the next, and how when you stopped working in Professor Falk's lab and the papers were all with your name as the primary author, the direction changed entirely. And what was the catalyst for these types of things?
Well, I think what you say is probably characteristic of the spirit of science, because one of the reasons for choosing science as an exercise or as a profession is, I think, that you can never predict where the path will take you. And the rule is always not to take the broad roads, the easy routes, but to seek the, of course, the unknown, and also not know what's around the corner. And then it's important, I think, for the pleasure of science to catch the leads that you find, so that you have to be the observant. You have to somehow let your discoveries or observations lead you further. And the kinds of changes in my own work that you pointed out is highly accidental, and not even in retrospect one can make that sound logical. The main reason for me and my close partner at that time, Ulf Stenevi, to get into neuroplasticity and regeneration was highly accidental through the visit of Bob Katzman, who's a distinguished neurologist here in the States. He was on sabbatical leave in Lund, and at that time he was studying Parkinson's disease, and the experiments he wanted to do was related to sorting out the mechanism of action of L-DOPA in the brain. And for this purpose he used standard lesions of the dopamine system that we were working with. And by looking at specimens where we had been looking many times, he immediately picked up that there was something highly interesting going on, namely tremendous sprouting and regenerative capacity that where regeneration over millimeters took place. And he pointed this out to us and told us why this was very interesting, put out some key references for us to read, and that made us realize that, oh gosh, this is something tremendously interesting. And that's made at least my work change direction completely. When it came to neural transplantation, that is more, I think, a case of getting inspiration from outside. In the beginning of the 70s, there were some groups, notably 2 groups, that started to report on neural transplants in neonates and in the NTI chamber. One was Gopal Das, who was with Altman at Purdue, who published a paper in '72, which I know I and Ulf Stenevy picked up and found very interesting. They put neonatal cerebellar fragments into the cerebellum of neonates and got survival integration of the neurons. And this gave us the idea to try similar techniques, but in adults instead. Similarly, Lars Olsson and Åke Seger in Stockholm started to work with grafting into the NTI chamber. And so that the kind of neurons we were interested in, the dopamine neurons, the noradrenaline neurons, survived very well, grew tremendously, and we tried to apply that to the brain.
What made you focus on the monoamine systems for transplantation?
Well, these were the systems we knew well. We had the methods for this. This is another key factor that we had at that point, in the beginning of the 70s. Very powerful techniques to study monoamine neurons in their entirety. Mm-hmm. And I think one of the key factors that made our grafting work, the one which we started in 73, particularly interesting, was that we had a technique to selectively visualize one subcomponent of what we grafted and be able to study over time the behavior of these neurons with a technique that stained them in completeness, the cell bodies, the axons, the terminals, the dendrites, the whole system.
Now, you say you started this in 1973, but the publication didn't come out till 1976.
No.
That's a long time.
That's right. It took us almost 3
3
years.
Yeah.
Yes, that's another thing that one can, or I can contemplate about, because it's clear that if we weren't 2 of us, Stenevy and I, who both sort of pulled in different directions in this work, we wouldn't have stayed on it, because we had such a number of failures, totally negative results. Mm-hmm. And if we hadn't, for some strange reason, been convinced that this had to work somehow, we would never have stayed with it. I think we must have done, let's say, 10 sets of negative experiments before we got anything to work. And the first kind of neuron we got to work for grafting was sympathetic ganglia, fragments of superior cervical ganglia.
So when did you realize this might be useful clinically?
I did not recognize this, or I didn't feel this was a meaningful idea until well into the 80s. Around 1985, Olle Lindvall and I came to an agreement that we should try to get the permission to do studies in patients and get the ethical regulations for it. But then we had worked about 5 years with the animal work.
Well, in 1979, your first paper showing behavioral changes after a transplant, which corresponded with Perlow's study with the same type of conclusion. I believe it was the concluding sentence was that it might be useful clinically. Yes. So you obviously had thought about it.
That's right. No, yeah, and I and Ulf Stenevi reacted very strongly to this blunt, pretentious claim because we felt that a pharmacologically induced or drug-induced behavior test, as was used both in our paper, parallel paper in '79, and their paper that came out at the same time, didn't justify any conclusions of this sort. So, and we at that time we had started a collaboration with Sue Iverson, who was the behaviorist in our collaboration, and Steve Dunnett, who was Sue Iverson's student. So we called them up and asked, isn't this hilarious to make this sort of conceptual leap? And they said, yeah, this is ridiculous. So we put together a letter, a correspondence note to Science, where we said that without experiments showing effects on the underlying symptomatology in these rats,
and the
spontaneous behaviors, on the parameters of relevance for the clinical condition, like akinesia and rigidity measures or motor function, was meaningless to even speculate on the use. They considered this note in Science for several months before they sent it back and said that they were not going to publish it. So we never got that discussion into Science then. This issue is as actual and relevant today as it was in '79. I think that it's still one of the critical points, that animal experiments cannot take you all the way. You can never predict from the animal data what will come out clinically. Then I must also admit that when the first clinical trials with adrenal medulla was done by Bjorklund, we thought this was also premature, and that one ought to have much more preclinical data. But it did influence the further course of events, because Olle Lindvall was then recruited to be the neurologist of the adrenal medulla studies, and when he decided that it was an ineffective procedure, he had, I think, prepared himself in a kind of psychological sense to attack this with the fetal tissue instead.
Well, in all fairness, though, at the time that Bjorklund did his first transplants, the MPTP monkey model did not exist, whereas when Madrazo did his, it did exist. So there was really a conceptual difference at that point.
That's true. I mean, now clearly the demands are higher because the possibilities to do good preclinical trials are much greater.
So if you were to point out your most distinguished accomplishments, what would you be most proud of at this point? Or is it too early in your career to say?
I think that for most people who are really hooked on science, nothing of what one has done is particularly interesting. It's always what's in front of you, or what is left to be done, and that seems always to be more important. So I'm not sure I can. There are some publications, for sure, that I think are better than others in my production, and some of the best ones are in the anatomical mapping field, where I think we've done some technically quite good stuff. And some is in the fetal transplant field, where particularly those things that are related to the methodological development, I think.
To change tacks a little, could you discuss briefly, I know it's hard to get into it extensively, but some of the possible mechanisms for how grafts work and your feeling about why aiming at a strategy of reinnervation is so important?
It reflects very much the way we got into the field as neuroanatomists. From the beginning, I've thought that this approach is a neuronal replacement technique, that neurons are lost from the brain. You try to find elements that are as immature as possible and see whether they can run through or carry out their developmental program in the environment of the lesion brain in order to mimic or behave as if it were the kind of neuron that's missing. So you have the nigal neurons, the damage, you lose the nigal striatal pathway, and ideally you would like to put the neurons back into the nigra and get them to seek their track back axons up to the striatum, re-establish connections, and make a new system. Now, quickly we saw that axonal growth didn't take place in white matter. This is something that more recently has started to change. One can see considerable growth along white matter under certain conditions, apparently. But at that time, it seemed to occur only in gray matter. So we had to choose the trick to put the cells in the target area rather than in the original position, the ectopic grafting. But we have not left the basic idea that it's the pathway that's reconstructed. That's also why in the first papers we have in the title reconstruction of nigal striatal pathway and another paper reconstruction of the septohippocampal pathway, because it was the connectivity that we wanted to go for.
Could you talk about transplantation as a technique and as a tool and its possible applications for basic science discoveries, as opposed to, I think, the way neurosurgeons are looking at it at this point more as a clinical tool?
Yes, neurotransplantation has its own life as a research tool to sort out mechanisms underlying both establishment of connectivity as well as mechanisms underlying functional recovery in situations of impaired function. It could be after lesions or in aged brains or after manipulation in the neonates or in development. And this is a field that is still in an early stage, and their refinement of the technique, because of the small size of the rodent brains, is of major importance to make this more precise, more refined. And perhaps the most refined grafting experiments that is being done now is in early development, where they try to mismatch, take neuronal precursor cells or dividing precursors and put them in the wrong context of development in cortex or in cerebellum, and see how they can, if they are, when they are determined, their fate, their phenotypic characteristics, and their connectivity. And those developmental neurobiologists have to some extent, led the way in a technical development. When it comes to the more disease-related problems, I think that neurografting has had a role in promoting development of better and more standardized animal models, particularly when it comes to long-term evaluation. Before neurotransplantation, a long-term experiment went over 1 or 2 weeks. When we started doing transplantation experiments, we made experiments over 3-4 months. Now one expects graft studies to run over a year or 2 years. And this means that stable baselines, dynamic changes over time is much more critical to establish. And hence the mechanisms of recovery has become much more studied and will remain so. There is much more to find out about that.
Could we take a cut right now? Thanks. I'm not going to. Okay. Now, clinically, Parkinson's disease has taken the forefront in the transplantation world. Why do you think that is?
Well, it's become a kind of test bed for neurotransplantation. For us, it was a very natural start because we were working with the dopamine system. And we had this particular interest, we had the techniques. At the time when this was started, it's also clear that the Parkinson model in rats was the only one that was worked up reasonably well. It was a well-characterized lesion syndrome. There was much of behavior test procedures to be applied. The biochemistry and pharmacology was well known. The disease itself can also be said to be well characterized. One knows pretty well what's gone wrong, and the L-DOPA therapy gives a reasonable background to select dopamine, the dopamine deficiency as a critical element in the symptomatology. And for us, this served a good purpose because we could then work on the dopamine substitution idea, saying that dopamine neurons are lost, dopamine is needed to be put back. L-DOPA is currently used, but L-DOPA after some time runs into trouble because it becomes unstable, gives fluctuations. And what's the problem? Well, it's because the neurons are missing. So if you can put the dopamine neurons back, you may also be able to substitute for the lost dopamine function. So this gives a good working hypothesis to build on.
Had your earlier work in the plasticity field with the observations about the plasticity of the monoamine system in particular contributed to the choice to pursue that disease process?
Well, it was the catecholamine systems that we looked at in terms of regeneration of deletion. And because of our techniques, also the first CNS neurons that we studied in grafting was the monoamine neurons, the catecholamine neurons and the serotonin neurons.
From the neurosurgeon's perspective, I guess there's two fortuitous side effects of this pursuit. The first is that the target area to transplant in is, although it's 11,000 cubic millimeters compared to the rat being 25, that's a very small target area compared to other diseases that affect much larger areas of the brain. And I guess we're having trouble filling up even that small target area because it's so big compared to the rat. And that's fortuitous. And I guess the other thing that's interesting in retrospect is the role of the monoamines being regulatory rather than carrying primary information and moderating the amplitude, so to speak, or the gain of the nervous system. And therefore, a one-way innervation can be helpful rather than having to rely on a complex circuit.
That's why one has to question the limits or where the limits for these techniques may be, because systems that depend heavily on connectivity and pattern information, for example, the visual system, will be much more difficult to substitute in a functionally relevant way than the monoamine systems. Spinal cord injury may contain a mix of these. There may be some driving systems or more non-specific systems that play a role, for example, in spasticity and setting tones in reflexes or setting tones in the autonomic nervous system. And they are more akin to the dopamine system, of course, in a way of functioning.
But for the complex behavior such as walking, which of course is the big hope, that would be a far more complex and difficult challenge.
That's right.
As long as we're talking about limitations of transplants, maybe you could address some of the limitations of any animal model as it relates to Parkinson's disease, as well as the limitations of upscaling from discoveries that occur in the rat going directly to humans. And I guess the context or the backdrop for this is the fact that these experiments worked so beautifully in rodents and in monkeys, and here we are now 5 years after the first clinical transplant was done, and in Parkinson's patients, we're still only seeing behavioral effects that are of therapeutic value, but that are far less than what would be seen in the animal models. And so I'd like to get your opinion about that.
I think it illustrates the dilemma or the limitation of the rodent models in particular. I'm convinced that rodent models have to be worked on much more to make them better and make them more useful in the clinical perspective. Because currently, the rodent models can be used to establish basic functional properties of, for example, a transplant, so that one can generate data on basic functional impact and basic modification of behaviors that can be quantified in the rat. But it's very difficult to use these data to predict the limitations when the same technique is attempted in the bigger and much more complex human brain. And in order to be able to do that better, one has to be more refined about the rodent testing to sort out as many robust measures of behavior as possible in order to generate profiles of recovery. For the dopamine grafting in the Parkinson model, for example, one has to be much more aware of the distinction between basic tests, for example, rotation, and the tests of more complex sensory-motor integrative behaviors that may predict much better the shortcomings of the grafts in the clinical setting. And this is probably just as important if one were to try the neurografting in Huntington's disease, for example, where the cognitive impairments and the progressive dementia might be more devastating to the patient than the motor problems.
One thing that has become very clear to me is that very, very small modifications in technique, such as altering the age of the donor by a week, or the size of the stereotactic needle, or how far apart the grafts are, very, very trivial appearing changes can have vast implications in the success of the grafting technique. And so I'm wondering what you think of the field now and how you see the next 10 years progressing. For example, in the kidney transplant data, there was no sudden year where the success rate went from 40% to 90%. It was really a stepwise progression, almost in a linear fashion, over about 12 to 14 years. And I'm just wondering what your observations are.
I think one can comment it in two ways. One is that you have yourself in your work been able to identify the critical importance of minor factors, or what might seem minor factors because they are details in a protocol. But this illustrates that progress cannot be made until one is, or unless one is very rigorous about establishing every single element in the technique. And the outcome is going to be the sum of all these. So weaknesses in how a protocol or how a method is being worked out will immediately come out in the end result. And since we are no longer going to be satisfied with partial results, now we need to look for more complete engraftment to get much more effective transplants, and also transplants that can improve the patient's condition in a more complete way, then this will become even more important. In the early stages of this, it has been enough to seek evidence that the technique is basically feasible. But once this has been established, then one has to put the requirements up on a higher level and say, well, that's not sufficient any longer. Now we have to put our demands higher. And the other lesson, since you mentioned this on the organ transplant field, I think there is something to learn from this also. It took them about 20 years to get any technique that would be justified as a therapy. And interestingly, the major changes, the first successful twin grafting was made in '54. Then in the early, and then they had a lot of failures with allografts. In early '70s, with the introduction of the first efficient immune suppression, the azathioprine, they could generate reasonably successful allografts, but with a lot of medical problems. They generated sort of a whole new field of disease that had to be treated because they had the survivors from transplantation. And that put the field into on a halfway level that nobody really wanted what to do with. It was only when cyclosporin was introduced in the late '70s, that is 25 years later, that the results were good enough to justify the use. And it's interesting that developments there was outside the actual transplantation field, in that case in immunology. Here in neurotransplantation, the obvious developments that will have tremendous impact on the further development of the field lies in cell biology, understanding of mechanisms to regulate neuronal growth, survival, differentiation, how to generate cells from precursors, and how to handle this very powerful cell material that one can store from.
So far, we're at a very interesting threshold clinically. So far, none of the patients that have received transplants when the patients have idiopathic Parkinson's disease have demonstrated more than a modest improvement. I think that would be fair to say. Whereas the two patients that have had by far the greatest improvement were the patients that had MPTP-induced Parkinsonism, or a toxic exposure leading to Parkinsonism. So the question is, do you think that difference is due to technical differences between the ways the patients receive transplants, or do you think that's due to an underlying difference in the brains between the patients, and then where do you see the specific modifications going in the next 5 years?
These are critical questions. I think one cannot answer them at this point. There is a risk that the underlying disease process is the difference. That is to say that the substrate, the environment these growths is functioning in, is not good enough to get more than modest results. And perhaps they won't be long-lasting. There is also the risk that the underlying disease process will undermine, so to speak, any graft effect. And then this will also affect the longer-term evaluation of the procedure. I don't think one can say that yet, but I think it probably means that the requirements of an effective technique in idiopathic Parkinsonism is higher than it would be in a toxin-induced one because of the sliding baseline. So, and the need to have a transplant procedure that builds in a safety margin. One has to bring the system up on a level where it can cope with also continuous decline of the underlying system. And we're certainly not there yet. This can also be a reason to say that perhaps the most important role of the Parkinson field is actually to serve as a test bed for the neural grafting technique into the brain, and perhaps with the logistics of serendipity and research, perhaps what will turn out to be the fruitful outcome might lie in a totally different application, but an application which has been reached because of the development that one can and the progress one can make in the Parkinson field. So who knows, the neural grafting may never turn out to be a useful therapy in itself, but I think one can be sure that it will be the starting point and the inlet to a full new world of applications, if you wish, for this technique.
Let me turn to Huntington's disease. It's a far more problematic disease from the point of view of transplantation, in that it's a far more rapidly progressing disease. There's not only cell loss of one small subgroup of neurons, but it's got multi-system atrophy. And also, it's not just loss of regulatory neurons, but actually intrinsic neurons that have primary information encoded in them. So therefore, it's somewhere between spinal cord injury and Parkinson's disease in terms of its complexity. Yet, so much of the animal data looks promising.
Mm-hmm.
How do you reconcile these
In
two
some
problems?
ways, I mean, when we review the animal data, in some ways they are stronger than the Parkinson data. And that is because the effects on spontaneous behaviors have been better assessed in that model, because there are tests which are more useful for that purpose. So looking just at the experimental results in rodents, one would say that this looks quite reasonable. Now, as you say, the disease is different from the rodent model, and this is a general problem. Nothing of what neurotransplantation can be applied to is really well studied in animals. Some models, of course, let's say spinal cord injury or a stroke, can be more or less identical as an insult, whereas the diseases we're talking about, Parkinson's, Huntington's, Alzheimer's, for example, they are not known in animals, and consequently are much more difficult to study. So there we're waiting for the more ideal models to work this up. Waiting for the more ideal models, particularly in primates possibly, I don't think this field can develop in a healthy way without limited clinical trials. But because the basic science will start to develop in its own way. There are so many challenging issues on the experimental level, on the basic science level, that the scientists are going to be distracted and dispersed on all kinds of problems. So unless one can set very firm goals related to clinical trials, one will never get the methodological development and the basic science development targeted enough. So the scientists need this feedback. The clinicians at the same time need to find good basic science partners in order to, if they are interested in getting into this field. Because knowing that it will take decades before any really final techniques can be said to be available, one needs to have good symbiotic relationship between the basic science and the clinical part. And I don't think clinicians will stay interested over a long time unless they can make critical tests in patients. That's the real test. Does this work in a patient or not? If it works, then it is encouraging. If it doesn't work, it's a challenge to continue.
On the assumption that Huntington's disease is a progressive disease with very few, in fact, no real major therapeutic options. A few centers have actually gone ahead now and performed transplantation of one sort of another with various obvious methodological problems into patients with Huntington's disease on a compassionate use hypothesis. In your mind, obviously you have the technical ability to take the tissue and transplant it. It's not a big deal from a neurosurgical perspective. What would be your minimum requirement scientifically before jumping into a compassionate use?
Well, I think that compassionate use of this kind of, let's say, preliminary scientific data is very regrettable and very unfortunate, because it can set back development in a severe way. It will also jeopardize the ethical aspect that this may put the whole field in disrepute. So even if it is tempting from point of view of, or based on compassionate reasons, to use even prematurely techniques that are certainly not fully developed, it is counterproductive both for the clinicians and for the basic scientists. And this is true for neurografting in all kinds of diseases. Huntington's disease, we think, is a good target for developing transplantation procedures for. But we know too little. I think for us it's the aspects related to the fetal tissue material, how to define the parameters, what to take, what ages can work, to be sure that what we isolate will have the right properties, and to get a minimum set of data to indicate that the tissue is functional and viable for its purpose. And this has proved surprisingly difficult so far. The telencephalic tissue is more difficult to get, and we doubt we have sufficient control of the dissection of that material.
And the variables. Yeah.
It's too variable, and we don't know exactly what to take.
Well, talking about compassionate use and early studies, maybe you could draw upon the American experience with the adrenal transplants in patients with Parkinson's disease, because there was obviously a vast difference between neurosurgeons here in America and in Europe, in that nobody in Europe signed on to the adrenal transplant program, and numerous programs in the United States signed on, and that is a tremendous difference. And I'm interested in why you think one community completely went for it and another completely didn't go for it, and what lessons were learned, and how we can avoid those mistakes in the future.
It's as if these communities were put to the same test, and they responded very differently to the challenge.
That's right.
And in Europe, I think it's true that both the neurosurgeons and the neurologists kept active, skeptical mind. For example, they read the Madrasa's New England Journal article in a quite different way. When I met Europeans who had read it, they immediately realized that this is an incomplete report that lacks a number of essential information, and thus you couldn't evaluate it. Whereas in the American community, there seemed to be many more who were satisfied by the face value. Many went down to Mexico and came back after a day or two and said that, well, seems that's nothing strange about that, so it's probably okay. And then they jumped on it.
Do you think it had to do with any one or two dominant American personalities that
it might have been.
log-rolled everybody into this?
We felt that, I'm not taking any pride in this, because we both, our partners in England and ourselves, we were all thinking the same way. We couldn't understand these data on basis of what had been seen in animal experiments. And as long as one did not care about establishing the technique in a well-characterized animal model, one would never be able to interpret the data anyway. So the lack of this correspondence would remain intellectually disturbing until it had been corrected. So in our opinion, the most urgent need was to generate the necessary animal data, which never really came about, or simply wait with the clinical studies. And we felt that when we expressed this in the state, there was a subgroup of neurologists and neurosurgeons that immediately picked this up or had just the same attitude, but others didn't seem to even want to listen to it. And this must have been almost a conscious decision on their part, saying that if we don't take a chance, if you don't have courage enough to try something unknown, you don't deserve the success.
Maybe it's the American entrepreneurial.
Yeah, that's, and I think it's true that some of those who set the tone, they had more this entrepreneurial attitude, and would thus not really have been worried about the scientific aspects of this. And I think that the only good thing about this whole upster is that it has generated a much more realistic attitude in the field. So now, when there is much more reason to have cautious optimism, then people are actually cautious in their optimism, which is very good. I think everybody knows now that if one is going to do this, one has to make sure that it's done well. And otherwise, it's not worth even trying.
As an editor of 10 journals and numerous books, how do you view the responsibility of the New England Journal for having published that the original report?
I think it's still
which was rejected,
ununderstandable.
by the way, by the Archives of Neurology before it was published in the New England Journal.
Yeah, I think it's ununderstandable. One reason may be that the editorial, which was written by a prominent neurologist, was uncritical, and perhaps they were unfortunate with the kind of advice they got, which is surprising for a journal that has such a standing, of course, and where requirements are high. So one would really like to hear an explanation. It's clear that anybody who reads the article now realized the weaknesses of the article, and why this wasn't realized by those who reviewed it at that time is a puzzle.
As long as we're talking about peer review, this is an issue at the heart of where we stand in America right now. Due to the funding patterns in the United States under Reagan and Bush, there has been no NIH funding allowed for clinical transplantation purposes. And that has had not only a financial impact, but also has created a tremendous stigma on the field from the financing point of view, and has taken the funding of this research out of the peer review format. And the analogy in America occurred in the field of in vitro fertilization, where the government said in the early 1980s that in vitro fertilization could be funded by NIH as long as each protocol was passed by the Ethical Review Committee of NIH, which they then disbanded in 1982, making it, of course, impossible to have funding of NIH, using NIH money of the field of in vitro fertilization. And then you got mom and pop corner stores with different standards of success, and one could have a 5-fold variance in the success rate of in vitro fertilization. A number of people would lose their entire fortunes trying to get pregnant and be unsuccessful. And it wasn't until the obstetric societies actually intervened to set up an external peer review system to make recommendations to government and insurance companies and referring physicians that the system actually got straightened out in the last several years. And we're sort of at the same point here in America. And I wonder what you think about ways to encourage peer review and thoughtful science in the future. Now, of course, we're in a new age politically with Clinton being the president, and hopefully this will turn around as NIH becomes more involved in the funding. But how do you see peer review as?
Yeah, I think you've summarized it very well. I think this is essential, not just in this field, but also to avoid developments that create much more problems. The idea of funding outside a peer review system or outside the influence of the scientific community will only encourage and stimulate developments that can be very problematic. That is the application of techniques that are totally without foundation to justify adventurous clinical trials of all kinds of procedures, just because the climate is such that academics and scientists, they are crazy. They should not be allowed to guide this. But the patients and the compassionate reasons should be leading the way. And if you just have the money, you're allowed to do anything. That is sort of the message if you don't watch out. And I think at this point, it's very important that the scientific community takes the initiative to structure the work, not to prevent things from happening, but make sure that the quality standards, the scientific value, and the value for money is maintained, and make sure that what's done in this field is done seriously with long-term commitment, with the input of sufficient expertise on all levels. And this could very well be done within the NIH framework, and it's a pity that NIH has sort of ducked this, that they don't see that they could play a very good role, not to tell people what to do, but to make sure that standards are kept high.
As a way of following up on the peer review, I went to a lecture on ethics of transplantation, probably the 10th lecture I've heard on this subject. And this lecture was completely different than anyone I'd ever heard. Rather than throw out the ethical problems and then discuss the rational reasons why those really aren't problems, such as the difference between the ethics of the Nazi experimentation and the ethics of transplanting on fetuses where informed consent has been obtained, as opposed to the Nazi experiments where there was no informed consent, and also where the patients in the Nazi experiments were suffered greatly, whereas in the fetal tissue transplantation field, the abortion has already been done, the fetuses are dead, and there is really no harm done to them, and it's also used for compassionate use to help people. Rather than go through and take both sides of these arguments and try to break it down analytically, what this person did is they gave only the problems, one after another, after another, after another, for 45 minutes. And whereas I've had numerous discussions even with fundamental religious groups, all of whom, to the great extent, have said, yes, we have no problem with the concept of taking tissue, and once it has been declared dead, and once the abortion has been done, we have no problem with using that for compassionate purposes, as long as we are assured that this is not contributing to the underlying thing which we object to, which is the abortion process. And so I've had even rational and good, healthy discussions with all sorts of people in that lines. But in this lecture, which was done in front of neurologists and doctors, including people in the transplant field, by the end of the lecture, they were hysterical. Absolutely, they went wild. And what became obvious is that there's a very deep history at the psychological level that relates to the history of alchemy, which goes back in mythology, and that the old alchemists believed that elements such as gold had more godly value than elements such as straw. And that if you were an alchemist and could convert straw to gold, that means that you were endowed with God's power to be able to transform. And the ultimate level of having God's power within you was to be able to make life where there is no life. And this myth is repeated over and over from the ancient alchemists to Rumpelstiltskin to the modern day stories of the Sorcerer's Apprentice, and even with Mickey Mouse in Fantasia. And the tragic flaw of all of these stories is pride. And I think that one of the greatest restraints that we all have to have as scientists is to make sure that not only we, but also our peers are not overwhelmed with pride and are truly putting compassion and the science first. Because I think that is the subliminal fear that is running through the Western history. And I'm just wondering what you think the role of the scientist should be in the ethics debate. Are we the ones that carry the ethical burden? Is it an external force that we listen to? Is it the role of society to set down the guidelines for us as scientists? Where do you think we fit into this?
I think it's probably unavoidable that scientists get into a dialogue with society and with colleagues and general public on these issues. My conviction is that scientists cannot operate without the actual support of the community we are working in. So whatever we do have to be consistent with the ethical standards, the morals, and views of things that are prevailing in our respective society. And it would be unhealthy to see it otherwise. So if the climate in the society, the attitudes are preventive, then the scientists should accept this. At the same time, I think that scientists are parts of an evolution as anybody else are. And I think ethical and moral questions are evolving just as much as any other attitude that people have. I don't mean that they would over time start to reduce their ethical standards or so. It's just that it's a kind of educational element, that understanding is very important for people's reactions. And it's probably healthy that the general public reacts in a negative or hostile way to things they don't believe they understand and they believe is potentially dangerous or can lead to consequences that they don't want to have. And this is a way to provoke the system, of course, provoke the situation, saying that unless you can convince us that we're wrong in this assumption, we don't want it. And then the scientists have definitely to be sensitive to these signals and say that, okay, let's try to explain how we see this, and let's try to make the science understandable. And it will always be a relationship, I think, between the respect the scientists can meet in what they say and what they stand for, and the understanding they can expect from people around them. So in the worst scenario, of course, the scientists will make procedures like the neurografting develop into a terrible mess that lacks responsibility and where irresponsible actions are perhaps more common than responsible ones. And then everybody will suffer, because they will not, then the responsible scientists cannot expect general public to accept what they're doing.
I think that if the same sort of pattern that happened with the adrenal transplants happens with fetal tissue, we could easily lose the type of credibility that is so necessary in such a obviously emotionally charged area.
And I think there is possibly more at stake, because neuroscience and clinical neuroscience will, over the next decades, develop new ideas that potentially could be applicable for neurosurgical or neurological application in therapy or in diagnosis. And interventions in the brain has these mythical and magic qualities that you were alluding to. Anything we do to our mind and to our brain can be said to be dangerous. We shouldn't manipulate the brain organ. And in a wider context, we shouldn't even try to correct what's gone wrong in the brain unless it's done with very crude means. If we don't handle the situation now well, because neural transplantation is one of the first pioneering techniques that is exploring the possibility to do something significant about restoration in brain disease. Then we may, if this is not handled well, and the respect the lay public has for the neuroscience community at large in its ability to handle these issues from point of view ethics and guidelines and regulations, then of course they are not prepared to give this community the confidence of doing anything else. So this will then just, the problems will magnify and become more and more difficult. If, on the other hand, the clinical and basic neuroscientists can be thoughtful about how they handle these new developments, interventions into brain function with new techniques, then perhaps we'll lay a very good foundation for future developments.
What I find interesting is that fetal tissue research has occurred for over 30 years, including the development of the polio vaccine, for which the Nobel Prize was awarded in 1954, mechanisms of viral infections, including the rubella virus and the vaccine for chickenpox, inherited diseases, diabetes, aplastic anemia, thalassemia, Fabry's disease, respiratory distress syndrome, sickle cell anemia, mechanisms for AIDS, epilepsy, Huntington's disease, Parkinson's disease. And it was not until the concept of neural transplantation and altering of the mind with fetal tissue research that all of this came to a head. And I just find it interesting that the combination of the subconscious fears that society rightly has about bringing these two areas together and the alchemy concept. It's just interesting after 30 years that this is the thing that precipitated this type of awareness.
Yeah, I think it's true also because one can make an analogy to a blood transfusion. Blood transfusion is cell transfer from one individual to another. And that's been accepted. Some object, of course, but by and large, people find that the most natural thing. And on another comparison, I don't think that neurografting in the brain is, on a principle level, ethically more troublesome than use of brain-active drugs, because any drug that is developed that can modulate or interfere with brain function is a potential danger for either killing the person or making him crazy or to destroy parts of the brain or to upset the mind in some other way. So misuse is not a new thing in this field. It's not that aspect of neurografting, I think, that is worrisome. It's the essence of the transplantation idea that has generated most of the problems, I think.
In terms of ethics, one of the main objections, I don't know how to say this politely, of the Swedish School of Research has been the transformation from the rodent work directly to patients. And that's been a real American perspective, given that all the monkey research up until Stephen Dunnett's work recently has been done here in America. so that's coming from my colleagues. And when Professor Bjorklund was asked by John Sladek right after the adrenal transplant experiments why you didn't do this in monkeys first, Professor Bjorklund said with a straight face, it's very simple, we can't get an informed consent from the monkey. And apparently he was reflecting on the ethical and animal rights groups and the pressure they have put on the Swedish research teams. And apparently, I guess it's very hard or impossible to do monkey research in Sweden. And I wonder how you think that has affected the course of science. And if we have anything here in America to learn from this.
Well, I think that the problem is to make a monkey chronically sick. It's not necessarily to do experiments on monkey. That's, although it's complicated, that's quite feasible. To make them chronically sick is likely to be a real problem to convince the committees of. Now, we have rationalized that situation and found our own strategy which justifies this by saying that careful use of the rodent model can bring us as far as we can experimentally in order to be able to justify clinical trials. And then we say that no animal model will take us farther than that, or farther than a certain point, because we are not studying the disease. The critical problems, the critical unknowns have to be tested in the patient. And I think it will remain so, even if I think it is highly desirable to have more work in primates, but they are bound to be limited, and they will never solve that dilemma that without an active testing of procedures in the actual disease, one can never evaluate the progress. We will never know how far from the goal we're standing and whether we make progress at all. Even if we make serious efforts to improve our techniques, how can we know that these are real improvements unless we can see improved results in patients?
So given all these variables, the multiple questions, the species differences, the social background, the different countries, peer review, what type of centers do you think should be doing this type of research? How many centers do you think are needed worldwide?
Well, first of all, it's clear that it's very important that some U.S. centers, strong U.S. centers, go into this. Healthy progress is depending on that. And that is not just to flatter U.S. science because it's very strong, but also the fact that the speed of development will not be sufficient to maintain an active interest unless there is sufficient body in the whole enterprise. I think, however, that there is an optimum somewhere. There should be strong centers. A strong center needs the combination of the neurosurgery, the neurology, and the basic science support. We are convinced that one has to work with non-invasive techniques like PET scanning in order to be able to do a good program. So that's also an essential element. And given those 4 elements, there can't be too many. We think that in Europe there may be 4 or 5 places. In America, perhaps between 5 and 10 would be good, but I'm not sure that more than that is needed as long as there is good coordination to some extent and exchange of information between the groups to help in progress.
With the adrenal transplant programs, what happened is virtually anybody that wanted to do it started doing it, with the only restrictions being the local institution review board for the particular hospitals. And it even got to the point where one private practice group in the Midwest was performing these experiments. Now, they actually did quite a good job. But the broader question is, what would you say to the vast number of groups that will obviously get into this once anybody has the first promising result?
Well, I would try to keep people waiting as long as they are not going into this as a scientific enterprise, the long-term scientific commitment. I'm convinced that we are not going to have an acceptable procedure for a long time yet. And an acceptable procedure has several elements. It has to be effective. It has to be fully reproducible and predictable. One has to be able to predict results sufficiently well. It has to be safe. One has to get away from the randomness of the current approach, where there is a clear risk of infection, of contamination, accidental problems of all kinds. And then finally, it has to be practical and justified as a therapeutic routine, which is also clearly not solved yet. And finally, it has to be fully ethically acceptable. And there we are struggling with the basic problems. How could this therapy, if it was a therapy, be based on a procedure that is ethically fully acceptable, as well as practical, safe, and reliable? And I think before we have reached that stage, we should not present this as a therapy. I think it's then remains research, and research should be kept within research programs that are run in a very rigid way.
So then where do you see the field of neural restoration? And I'll say restoration specifically because it may be that as neurosurgeons we're out of a job here in 5 years or 10 years, and it may have gone to a completely different level. Where do you see things in 5 or 10 years?
Well, I think that neural restoration...
Should I be trying to look into the field of neurology rather than neurosurgery?
No. My view of this is that we are just in the beginning of a development that will be tremendous for all parts involved. Also the basic scientists with the challenges that come out of it, as well as the clinicians. And I think we are in for a development that will open up many new avenues. It will partly depend on, let's say, the transplantation approach, how that can be used in various ways and finding ways of using it. But there are inputs from other sides also, gene therapy, for example, and the obvious application of gene therapy in the field of transplantation. Genetically engineered cells to modified cells in order to get gene and gene products into the brain, be able to use gene technological approach for therapeutic purposes. And there it's not just restoration, it could be tumor treatment, how to effectively kill tumors could be also based on transplantation approaches. And then there will be new knowledge coming in from identification of genes that play a particular role. For example, the recent identification of the Huntington gene is likely to have a very interesting impact also on the transplantation field. Perhaps in a few years' time, we'll be able to devise a particular variant of transplantation based on the insights in what is actually going wrong, depending on this genetic defect. Same thing maybe with the Alzheimer-related dementias, that this could also give a new way. Intervention into the brain will always have the same challenge, and their development of efficient cell implantation approaches will be one of the most powerful tools one can apply.
Let me get to an existential question for you. When I was taking my neuroanatomy class, Professor Powell of fame of mapping the striatal system happened to be a visiting professor at Hopkins working with Vernon Mountcastle. And he taught my section in the laboratory. And one day I was talking to him and asked him about what he thought about the Nobel Prize. And he relayed a very interesting story of one of his best friends who spent his whole life working for the Nobel Prize and the year that he was most likely to win it, he died. And he spent his whole life chasing it. And then I've seen other people who had won the Lasker Award, where, of course, there's a 50% rate of winning the Nobel Prize after winning the Lasker Award. And every year when the Nobel Prize is awarded and they're not there, they walk around the halls with their heads hanging down. You don't impress me as being that type of person. You seem to be driven completely differently by a very internal clock. What drives you? What makes you?
Well, don't you think that ultimate rewards of that kind never can satisfy the curiosity and the kind of almost passion-driven mechanisms that characterize most scientific work? I don't think that it's fair to most scientists to say that they are chasing around for discoveries just to be able to compete for a visible reward of some sort. I think these are quite different sectors of life. Most people wouldn't object to a sum of money as a prize or whatever. Of course, that's appreciated by everybody, but it can never replace the motivation that is driving the scientists. I think what I've heard, what individuals report as the most disturbing aspect of public appreciation or visibility is that it disturbs work. They say that this interferes all the time with what I want to do, and I'm sure this is obvious to most scientists. I think I'm bitten by what I would think is a good scientific bacterium. I have this infection, and I don't know what it constitutes, but it's probably the same thing that drives a neurosurgeon to become dedicated to his work. Or
my roommate in college won the Olympics in foil, and right before the Olympics, he had a recurring dream that there was a long tunnel with a light at the end, and as the Olympics came, it got closer and closer and closer, and then the day before the Olympics, he realized that the light was a door. And the day that he won the Olympics, the next night he had a dream, and he opened the door, and there was nothing there.
There's no light then.
And what he took from that was that it's just a transient goal, and it does not define the rest of his life. And he has since gone on to be a virologist at the Karolinska Institute and studied with David Baltimore, and has a completely separate career after that. And
I think the happiest situation is not for anyone in our field is to maintain the curiosity where every new twist is potentially serving you with new surprises. This is the nice experience that one would like to maintain over time in science, that curiosity is never fading. You find new discoveries or new things around the corner that are just as surprising. Actually, the most enjoyable things is when things appear a bit different than you thought they were. To put the goal in one basket and say that this is what we're going to crack must be to ask for trouble. I mean, then what's left once you've supposedly solved that problem? Well, then you must quit somehow, do something else, and that's nothing to look for. So, of course, in this science, the issue is not necessarily to find a particular effective method for a particular purpose. It's the biology and the mechanism and the understanding that is the important thing. And that can keep us busy for many lifetimes, I'm sure.
They say one of the hardest things for a scientist to accept is somebody else's vocabulary. Yet you have personally defined 90% of the syntax of this field. How does it feel to see thousands of papers now published using your vocabulary? to look down at,
Well, I don't
at that and
well,
see that type of impact.
yeah. I don't think I can see it that way. I think it's such an evolving. I think I've always felt very much for seeing this field developing in a nice, healthy way. And I've always thought that the most important thing is that it has a good soul. The scientific soul of the field should be healthy, dynamic, creative, and friendly. I think that's perhaps what I've been most pleased with so far, that this has become a sector of science which is enjoyable. Good people with the right interest and the right minds have chosen to go into it. And this is what one would like to maintain, to make this also an intellectual enterprise that is worthwhile. And then the rewards are immediate, I think.
Well, for those of us that are younger, you've been a tremendous role model scientifically as well as a modern-day alchemist. I'd like to thank you very much.
Well, thank you.